once we get through to the point and show that we can have an effect on a disease, we can actually, hopefully really make a difference by having a whole field of people working on making different bacteria that work on different cancers.
This is the Discovery Files podcast from the U.S. National Science Foundation. I'm Nate Podker. The National Cancer Institute reports that over 2 million Americans are diagnosed with cancer annually. Statistically, more than 600,000 of those will lose their lives to the disease. As American innovation continues to push the boundaries of modern medicine, we're joined by Neil Forbes, an NSF-supported professor whose group develops molecular treatments for some of the deadliest cancers, innovations that could be transformative for Americans living with cancer. Professor Forbes, thank you for joining us today.
Yeah, thank you for inviting me. This is quite an honor.
How might bacteria, salmonella in this case, be used to fight cancer? This seems like it might be a confusing thing for a general populace.
So bacteria have a lot of abilities that a lot of other drugs can't do. First of all, when they're administered, they accumulate in tumors at ratios over 1,000. Sometimes we have some strains of bacteria that accumulate in tumors more than 1 million times more than other organs. This allows us to both make them much more effective and much safer. So we can deliver drugs into tumors that would be toxic in other places, but are only effective in the tumor because of that tumor accumulation. There's another thing that they can do. So salmonella particularly, which is what I've been working on for over 20 years, has an interesting physiology that allows us to deliver drugs in a way that's very unique. Everybody knows that salmonella can cause problems in your intestines. And one of the things that it does is it invades into the cells that line your intestines, and it does that to hide from the immune system. This is not a good thing when we're dealing with when you've ingested too many of these bacteria. First of all, the bacteria we're using have been made completely safe. So we've removed all of the endotoxins that cause them to be toxic to humans. But we use this facility that they have to get inside cells to deliver drugs inside cells. So a lot of drugs, both on nanoparticles, liposomes, even just regular drugs, get taken up into cells into something called endosomes. And cells don't like having foreign material in, so they process them to something called a lysosome, which degrades it and eliminates them. Salmonella don't want to be eliminated when they're inside cells. So what they do is they inject little proteins into cells that make them form into something called the salmonella-containing vacuole. The people that named this weren't very creative. Those vacuoles don't become lysosomes, but they stay inside the cell so that the bacteria can stay there for long periods of time. We don't want the bacteria there, even when we're treating cancer. What we want is our drugs that the bacteria are making to be inside the cells, inside the tumors. So once the bacteria have invaded into the cells, we've made a genetic circuit that the bacteria, using their own innate abilities, sense that they're inside this vacuole. They break open and they release their contents into the cancer cell. So this gives us a double layer of specificity. The bacteria have accumulated in the tumors, and now we've got them to invade inside cells. They've delivered their contents, and that contents is only inside cancer cells in a place that we can't get other drugs. So all of those markers that we've spent the last 40, 50 decades researching that are inside cells we have a really hard time getting to, we now have a way to get to. And not only that, we can get to them only in tumors. But bacteria in general have the ability to do many of these really cool tricks that we just can't do with small molecules. We can't do with antibodies. We can't even do with modified T cells like CAR T. We can modify them completely, make them work like I wrote in a paper a long time ago in 2010. We can think of those little robots that can be manipulated to do tasks that we couldn't get to make in any other way to solve problems that we really need to solve in order to make cancer treatable and curable.
So I'm going to attempt to summarize that. Bacteria, salmonella specifically, naturally go after certain areas, and in this case, they'll target the cancer itself. And you have engineered them to deliver the therapeutic drug and kind of self-destruct when they get to the cancer.
That's exactly right. Yes. So that self-destruction has another benefit, of course, is that every time that they invade, they self-destruct, making them much safer. So they're like little targeted. You can imagine them like little targeted payloads, a little go into cells, and then they burst. And so they're no longer living bacteria inside your cells, anywhere inside your body, after they've delivered the drug into the system.
I can imagine that being a lot safer and better for the body than something like radiation treatments.
Right. So the idea is we can make it a lot safer than radiation because we can get the drug right to the spot we want it to, and we don't have to worry about affecting other tissues around it, which is one of the big problems with radiation, is that it affects the rest of the body.
How did you originally get interested in the therapeutic potential of bacteria?
Well, it's an interesting story. I was working in a lab studying blood vessels and oxygenation, and we were looking at why do cancer cells, are they more aggressive when they don't have oxygen, or are they less aggressive? And this was a big question at the time. And I was going to do a lot of very difficult experiments to figure this out. I asked a question to one of the most famous people in the field. And I said, okay, if I answer this question, it makes them more aggressive, less aggressive. We could make drugs to work on those methods, and we could find a way to address that. But I'm a chemical engineer by training. If oxygen can't diffuse to those locations that are anoxic or hypoxic in tumors, how are larger molecules, biologics, antibodies, how are they going to get to that site? And he didn't have an answer. And it led to this frustration, which is like, we can do this work and we will never be able to make a drug to target that problem unless we find something that's not limited by diffusion. So there were other people in the lab kind of working on a bacteria-related thing. And it occurred to me, again, as an engineer, that they're not limited by diffusion. Bacteria accumulate energy from their environment and actively penetrate through tissue, which in my early work, I did this, 15, 20 years ago. We actually measured and quantified that ability of bacteria to actively penetrate through tissue to overcome those diffusion limits that exist within tumors and cause them to be difficult to treat.
So a good example of how you have to do the building blocks to get to the eventual place you've landed.
This is something that's taken a lot of pieces and a lot of time. There was that piece of the fusion piece, and then the accumulation in tumors components, and then the intracellular delivery. And then from there, my lab is now very focused on immune stimulation because of the excitement that people have had about immune therapies, but then also that they don't work on a lot of patients. So how do we make therapies that can be functional in patients that don't respond to immune therapies, but still trigger the immune system, which is, in my opinion, probably one of the best ways, the best chances we have to get curative therapies for some very difficult to treat cancers.
Like you mentioned it in the intro, but can we tease out a little bit how you're modifying the salmonella for this treatment at this point?
One of the big modifications we have is that genetic switch that has them lyse once they're inside the cells. We call this PSCJ lysine E, because that's the promoter that we use that has them sense when they're inside cells, and the lysine E gene that causes them to break open. We have other couple of modifications that we have. We've modified them. We discovered in 2019 that they need flagella in order to invade into cells. So we took one of the master regulators that control both flagella and the production of those needles that I talked about that inject things into cells. And we put that under a promoter that responds to aspirin. It's actually salicylic acid, which is a metabolic product of aspirin. I won't get into the details of this, but if you're taking a normal human dose of a single human dose of aspirin would cause the bacteria to go from being not have flagella to have flagella. Why is this important? When we first inject them, that flagella causes an immune response, just like the other piece that's been taken off, the lipopolysaccharide, the LPS, which causes an immune response, making them much safer. But it means that when they get to the tumors and they start to grow and replicate within the tumor, we can give a dose of aspirin, and now they are able to invade into cells. So we have them so they're basically inert and safe. Once they're in the tumor and have been cleared from the blood, cleared from the other organs, we now can give a dose of aspirin, they invade into cells, and now they deliver their therapeutic.
It's interesting that a safe drug such as aspirin can be the trigger to make this process happen.
Exactly. So that was actually why we picked it, was because it's already approved. We wanted something we didn't have to make the regulatory process more difficult. And we already knew that there were bacteria that responded to salicylic acid. So we were able to kind of put the two pieces together. And we've actually demonstrated that all of this works in animal models. So we can show that we can control delivery with all of those pieces.
You've had some success testing this out now, and I believe you've done animal trials and are getting ready to head into human trials. Can you talk a little bit about the results so far?
I should talk about, maybe there's multiple different ways you could use this. We both think of this as developing specific drugs and specific cancers, as well as being platforms. So you can, what I've described so far describes a platform that can be used to deliver many different things. And maybe I'll talk a little bit about some of the things we've thought about delivering. The first one is kind of obvious. We made the cells of cancer cells avoid cell death. It's how p53 works. It's how a lot of different well-known pathways that lead to tumor formation function, is that they turn off their ability to die when things go wrong. But there's a single enzyme that controls that. There's a whole cascade, but there's a single executioner, what's called a caspase. And there's many different caspases, but the key one is caspase 3. So we actually delivered caspase 3. Now, caspase 3, in order to be part of a whole network, has to respond and only turns on in certain environments. And that's how cancer avoids it, by not turning them on. But we actually just directly inject activated, fully functional caspase 3 that a colleague of mine, Judy Hardy, here at UMass in chemistry, developed so that it is always active. This would seem like this would be really dangerous. And this is where the bacteria come in, is that because we're only delivering caspase into cancer cells and it has to go inside the cells to work, caspase 3 allows us to only kill cancer cells. And the interesting thing is, like radiation and chemotherapy, once we deliver caspase 3, the cells die, and it induces an immune response by having that cell death present inside a tumor. We get activated macrophages, we get more influx of T cells. The first product that we have is this caspase 3 delivering salmonella. And we're at the point we've had our initial meetings with FDA, and we're talking with funders, and we're looking to get to clinical trials in the next few years. So we've seen complete eradication of pancreatic tumors with this therapy and liver tumors. And we've also shown that it dramatically reduces the growth of metastases, of breast metastases. So we think our first indication that we're looking at in human trials is liver cancer, because we feel there's an important unmet need there, that there's not very many therapies available right now for liver cancer. So we're working on that. But once we've shown that it's effective in liver cancer in people, I'm dreaming a little bit far ahead, because we've seen how effective it is against late stage breast cancer, which also is deadly and there's not many therapies for right now. We're hoping that once we get it approved and available for liver cancer, that we can move into metastatic breast cancer.
Specifically, right now, you're targeting pancreatic and liver cancer. And these are, I think, generally speaking, two of the most deadly cancers people can get. Like,
right.
Something like 10% on a 5-year timeline.
Yeah, it's about 20%, but still it's being diagnosed with pancreatic or liver cancer are very fatal diseases. And because they're not detected very early, and so we need drugs that work after they've spread. One of the other really cool things about bacteria is that they accumulate in those metastases. So if you have, say, liver metastases that have spread to your lungs, often at this point, you're on hospice, and then because there aren't any therapies once it is spread like that. But bacteria accumulate in those metastases, and we have some really wonderful data showing that, and showing that we can actually reduce the volume of those metastases once they've accumulated in those small secondary tumors and deliver drugs just to those regions. We get a ratio, we see about 850 times more bacteria in a lung metastases than the lung tissue that surrounds it. So they really home into that environment where they're not cleared by the immune system, which protects the normal tissue. You kind of talked about it right at the beginning. Salmonella, and everybody knows it causes, intestinal diseases and diarrhea. It does. One of the things that I had a meeting 10 years ago with a patient who went to a scientific meeting, and he I can't remember which cancer he had, but he had a cancer, and he was trying to find out what to do to treat him, how he could treat himself. And I asked him, this is an N of 1, and I understand this is an N of 1, but I said, I'm working on bacteria, on salmonella. Does that scare you? He said, no, the fact that it's dangerous makes it wonderful. This is why chemo works, this is why radiation works. You want something that's powerful. You don't want something that's like weak and may or may not work. It's like, if you're going to treat me, right? This was someone who was facing death, right? His own death, his own mortality, and thinking, don't dance around. Make something that works. And if it's going to have some side effects, this is the difference between treating cancer and treating other diseases. If you've got a cold, you don't want that, right? This is something we have to understand. And there's a difference between treating like early breast cancer detection. You might not want to treat this way. This is for people with late stage, the people where, like you said, when you have 20% 5-year survival, the side effects from bacteria may be things like, headaches and fever and runny nose, a bloody nose. I think that you don't want that if you don't have many other symptoms in your life. But if you're thinking the chance of me making 5 years is very low, though all of a sudden those symptoms look very, very different. Those are the symptoms, like I said, I mentioned this again, that was the predecessor bacteria, and that was the symptoms they saw when they put those into humans. So we've seen this before. We know that they're mostly safe. there's lots of people working on making bacteria safer. And as you can see, we've made it safe and we've tested in animals. We know that it's safer. And I've described some of the mechanisms by getting rid of the flagella, making sure that they lyse. But there's other things we can do to make it safer. But the point that I really want to emphasize for the public and for everybody, if I can say this is worth doing. It sounds scary, but the scary is actually why it's worth doing. It's not very well known. I've been working on this for 20 years though, and there are other people working on this, and we have a huge potential. And I think that rather than saying it's not scary, the message that I want to say is we should use the strength of these organisms to benefit human health. The more interest and excitement that people can have about this, the more it will lead to people asking, hey, how can I get that? How can we get more funding? How can we get more people doing this research? I think the potential is just is so great that for me, some people are scared of having competition. I'm not scared of competition. I want to see as many researchers getting into this field as they can. I want to see, I don't know if you could hear this. In my vision, there are so many different things that can be done. I can't do it all. And I don't want to do it all. If we could attract more people to work on this, I think we could really make a difference in health. I'm not saying this is the way. We don't need to take away from other strategies. I just think that we can attack diseases and solve problems that other things can't. And I don't want to miss out on the chance to make a difference in health by people being concerned or scared of bacteria. It's not because they're not scary, but it's that we can control it and we know how to make them safe. And with more work, we can make sure that they're even safer. And I think that with that, there's a lot of potential that I don't want to miss out on.
So I want to talk about the path to market a little bit right here. So you mentioned the trials, and there's a lot of FDA issues that have to happen as things get developed. But part of this process, you have developed a company that is spun out of your lab that is working to bring these things to market. Can you talk a little bit about Earnest Pharmaceuticals?
Yeah, so Earnest Pharmaceuticals was a company that I founded with a couple of colleagues of mine. We started in 2016. And through a lot of support by the NSF, which I'll talk about later, this is developed we had early funding to help develop these products. And the goal of commercialization is we don't want to just be academics. We want to have the whole purpose of doing this work is to make a therapy that can help people. And to get it to the market, it requires lots of clinical trials, lots of testing, which is very expensive. And this provides an opportunity for investors and people to gain value. So we have to form a company, have to get investment, and in order to do clinical trials. So the next steps are in oncology, clinical trials are a little different than other drugs. It is unethical to just do a pure safety phase 1 study. So you do a combined phase 1, phase 2A, where you look at a little bit of efficacy in a small cohort of patients. But in that, you're also going to demonstrate that it's safe. So that's the first thing we're going to do, show that it's safe to administer to people. And I want to point out at this point that we've actually done the critical animal studies that FDA asked us to do to show that it's safe. So we've already done those studies, showed that the bacteria we're using are 50 times safer than the predecessor bacteria that we're using because of those pieces that I told you about turning off the flagella, and the fact that they lyse after they deliver makes them quite a bit safer. So we've shown that in animals. Now we're going into humans, and we want to, obviously we're very excited about seeing how well it works in humans. That's the next step.
And I think it's a good example of kind of the timelines that are really involved in bringing something from the tabletop study in a lab to market. This can be decades sometimes.
Right. So this is we're kind of a little bit into this time scale, but it's often about 10 years is what you look at, is to go from. Now that doesn't mean, a large phase 3 study will have hundreds of patients on it. So a lot of people could benefit from it by once that larger, in that case, that's the second study we'll do. But once that larger study starts, we'll be able to hopefully benefit a bunch of people. But it will be very controlled, because obviously we want to get good information so that we can then write the final acceptance package to FDA so we can market it and sell it to the public.
You mentioned that NSF funding had a little bit to do with this company getting off the ground. Can you talk a little bit about how NSF support has impacted your career?
NSF is really has an SBIR, so a small business funding program, and the NIH does as well. So early on, we had these partners between the company that we founded and which was run by a postdoc from my lab. And so I'm just a scientific advisor for the company, and we work very closely together. Those early grants helps us to develop the product and do things that academic grants don't do. They want to see that we can show that it works, the basic efficacy, but like actually developing it. So like making that flagella switch, the pieces that are going to make it so it's going to work much better in humans was developed using that grant funding. And then the next step, we received a phase 2 NSF SBIR, which is a larger amount of money, and it helped us to do some of the early CMC work. CMC is the how do you produce it? How do you make it? So in order to get to trials, we have to produce it using something called good manufacturing practices, GMP. And so we've developed that protocol for how to make the bacteria, and how to make them relatively cheaply, and how to make them at large scale. So we know how to do that. At the moment, we're just getting the funding to make those batches and to run those trials.
I know you had some grants before that also. How did that set the table for how you've developed things in the lab?
I talked earlier about how we can see bacteria penetrate into tissue. That was done using this device that I developed using NSF funding. So I was very interested because of my earlier work on how blood vessels related to tissue. So we made a microfluidic device where we had a flow channel next to cancer tissue. And we developed some of the key understandings of bacteria allowed us to do this. Although we've also used it for looking at how drugs diffuse into tumors as well. The bacteria, we could see them penetrate into the tissue and then grow. And so some of that early knowledge about how that happens worked with that device that the NSF supported us. I want to add one other thing. Since you brought up NSF, here at UMass, I'm part of something called the Institute for Applied Life Sciences. And the goal of this institute is to support the exact thing that you were talking about, that translation from lab research into something that's actually going to affect the world. And a lot of biological work, of course, is going to affect human health. That's what a lot of us are working towards. And we have something called an NSF-ART, which is supporting that translation from research to use. That also has supported my work. In fact, one of the things that I am very indebted to is that whole institute, which is supported by the NSF, has done. You can imagine starting a company. There's a lot of pieces that professors don't know inherently that I've had to learn, like how do you write a business plan? How do you talk to investors? How do you write, a product, a TPP, which is a product profile? Like how do you start thinking about how do you take this thing that we've learned? We know bacteria do this neat thing, but how do we actually make it into a product that could be used in the clinic? And a lot of that has come through NSF-funded support.
A little different mindset, like thinking about the business end of the world versus figuring out how things work. Like it's different.
I feel very privileged in order to be at that intersection of the two of those. I now teach a course on entrepreneurship, and one of the key pieces, I won't talk about this very much, but one of the things that's really important to me is that when you ask young scientists, and I say young, I mean not that young, people who are late stage graduate students, 2 or 3 years away from their PhD and postdocs, people who've done some work and really thought about it. And I say, okay, that's really cool what you're doing. How could you use it? About a third of them say, you would do this, 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 and this. They know exactly what it is. But a lot of them say, I don't know. And you say, well, is the thing you're doing directly useful? And they say, not really. Could you change it? And that thought of, I'm doing very, really cool things. If I just modify it just a little bit so that I'm focused on how it actually be useful, it makes it so that research becomes much more tangible. And so I think trying to talk to students, researchers, postdocs, early PIs about how to transition would make it so that a lot of our research has the potential for translation when it's successful. And it takes that really successful research and makes it that much more impactful.
And I think this speaks to the ideas of workforce development, where you're really setting the people thinking about the positioning of how they're moving forward from just research, like the raw research of learning how to do something or studying something or becoming an expert in something, and how that might manifest out into the world.
Right. And the other kind of ancillary benefit of all that is really exciting, that once you can say, hey, this is going to help a lot of people get excited and they want to hear about it. It makes it easier to talk about what you do because it relates to the public. People want to know that you're working on pancreatic cancer. People are scared of that, and rightly so. And when you hear about people who are well known who have pancreatic cancer, it's worrisome. So we need therapies to work on this, and hopefully increasing awareness of cancer, and that the research that we're doing is going to have a direct effect on people. I think will show the importance of why research is really important.
What's next in the next few years for you?
We can deliver whole viruses into cells. How would you want to do that? Oncolytic viruses, just like salmonella, there are viruses that, in fact, for example, the one we're using is a rat virus. It doesn't even infect in humans, but it grows in cancer cells and it kills liver cancer. Someone figured that out, and it's very, very neat. It also, once it kills the cells, it induces a massive immune response to happen in the tumor. And there's a lot of excitement about that. But one of the problems with oncolytic viruses is that they work really well on melanomas, so topical, where you can apply the virus to the skin. But if you inject the virus into the blood, it's often cleared very quickly because our bodies don't want viruses in them. So they get cleared. So one of the things that we thought is, what if we had the salmonella take the virus into the tumor using all the tools that I just talked about? So we put the genome for H1PV. It's a parvovirus. I won't get into the details of that, but it's small enough that the salmonella can carry it in. They then go into the cells, break open, like I talked about before. The virus is then the virus genome, not the whole virus. It's just the DNA that tells a cell make virus, which ordinarily you wouldn't want. But now if it's just in cancer cells, the cancer cell itself makes the virus replicates, makes a few million copies of itself, breaks that cell open. But that's not the main thing that we want. We've just killed a couple of cells at that point. But now we have virus instead of being delivered from the blood and going through your whole body and hoping that a few get into the tumor, now the source of the virus is coming right out of the center of the tumor, spreading just to the tumor. And remember I said that H1PV doesn't grow in normal cells. It only grows in cancer cells. So it's safe because it's only in the cancer cells. But it's replicating within those cancer cells and killing the tumor from the inside out, and then drawing in a huge immune response. So we get activated T cells, activated dendritic cells, activated macrophages. So we get both an innate and adaptive immune system. For the people who know that, that's a really important thing right at that site. And so we actually saw the thing this is why I'm excited about this. I had to get to all of this, is that we saw some tumors being completely eradicated with this, which we'd seen before with the caspase. But we took the immune cells from those mice, transferred them out, put them into new mice. And then we'd had some mice that didn't have T cells from actually they were full splenocytes. So all of the immune cells from the mice that were treated and some from control mice. And then we implanted those with the same tumor that we'd seen before. The control mice, the tumors all grew. And in the mice that received the immune cells from the treated mice, all the tumors, nothing grew. Nothing grew. What this means is, so those mice had never seen salmonella, had never seen virus, and never seen tumors before. So those immune cells had communicated to those new mice, tumor is bad and we're going to eliminate it. Why is this exciting for humans? What this means is if you treat a human with the tumor that you can see with a bacteria and a virus, it would train the immune system against that tumor. So if you had it recur or you had it spread to metastases, immune system would now treat it and reduce it. So it's producing a systemic anti-tumor immune response. And in my mind, there are still pieces for the experts out there in immune, that there's adaptions and mutations that are going to make this so there's not as wonderful as all that. But this is going to be one of the ways we're going to get towards treating late-stage cancer and eliminating metastases by making these systemic immune responses against those tumor cells.
Yeah, it's like you were talking about with the students, where you're looking at a piece and you're assembling how it might be pivoted towards a different project. And like I can see this, even the different treatments being effective for people that respond differently, because sometimes you have things that don't work that work for most of the population.
We have a couple of steps. And so you said next few years. And so next few years, we're hoping to develop that virus therapy further. We're going to work on different viruses. At the moment, it works on pancreatic and liver, and we want to expand it to other. Obviously, you can see why we did pancreatic and liver. These are very important diseases, but we want to be able to show that we can do other diseases. So we're going to have to expand to different viruses that work different in different tumors. Okay, that's the next few years, and working on getting the caspase into the clinic. But I have, those dreams. Just like you said, you could imagine that I'm working with a colleague. We're making a radiation responsive bacteria so that we can actually get focused radiation therapy into tumors. And then we can also then image them. Why is this important? If we know how an individual accumulates bacteria, we could make a therapy that works for that specific person. And you can imagine, instead of current personalized therapies where you have to take cells out and you have to do a lot of genetic engineering, you can imagine that the oncologist could have a shelf with one or two, as I've just described. You can imagine I've just described 3 different bacteria that you could use. You could have a shelf with 5, 6, 7 different kinds of bacteria, plus the biologics we already have. You could start doing some imaging and seeing how people respond to different bacterial therapies. These are obviously patients. So these are cancer patients. So this is the risk of that is very, it'd be well worth it for someone who has late stage cancer. But then we could then, instead of having to do genetic engineering, and the cost of CAR T, which is a fantastic therapy, by the way, but it's just very difficult because you have to re-engineer the cells for every person. We could have ones that are on the shelf, and once you've diagnosed the patient, you could be like, you're going to be best for virus therapy. You're going to be the best for caspase-3 bacteria. Or this combination works best if you hit first with caspase, then you hit with immune checkpoint. And we could figure these out so that we could now have a tailored protocol for different kinds of patients. And so one of the things I think is really amazing about bacteria is they're so modular. We can make bacteria that do multiple things and put them together. And in a way that you just can't with small molecules. They get big and difficult. We can make bacteria that do many different things, or we can mix them together or put them at different times. So I'm excited about what we could potentially do with this. That's looking decades in the future, but what we could do decades in the future. Once we get through to the point and show that we can have an effect on a disease, we can actually, hopefully really make a difference by having a whole field of people working on making different bacteria that work on different cancers. So I have a new collaboration with some folks that are working on like wound healing, colitis and Crohn's disease. So there's already been some work, there's been some clinical trials on that, but they have shown that you can increase the rate at which wounds heal. You can fight lung infections with bacteria. You can treat intestinal diseases. So I have this hope that soon we will be able to not just, I'm going to focus on cancer. This is what I'm going to do. But there are going to be people who are going to take this and we're going to make a whole arsenal of ways of targeting different human diseases using genetically modified bacteria.
Special thanks to Neil Forbes. For the Discovery Files, I'm Nate Podker. Please subscribe wherever you get podcasts. And if you like our program, share with a friend and consider leaving a review. Discover how the U.S. National Science Foundation is advancing research at nsf.gov.